Beautifully.
ऑडिओ व्हिडिओ थोडा
हळणर साहेब नमस्कार.
नमस्कार 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 सर.
नमस्कार. नमस्कार. कशा आहात?
बस मजेत आहे सर. तुम्ही कशा आहात?
मी आनंदात आहे.
हा
तुम्ही वेळ दिला आमच्या वेबिनार साठी. खूप खूप आभारी आहोत.
आम्ही पण तुमचे आभारी आहोत. तुम्ही आम्हाला अपॉर्च्युनिटी दिली की आम्ही काही आमचे विचार आणि थॉट्स तुमच्या समोर मांडावेत तसे.
आजच्या एज्युकेशन सिस्टमची अशी नीड आहे की इंडस्ट्री मधली माणसं, इंडस्ट्रियलिस्ट तुमच्यासारखे एज्युकेशनच्या फील्डमध्ये त्यांचं असोसिएशन पाहिजे, त्यांचं मार्गदर्शन पाहिजे. करंट ट्रेंड्स इंडस्ट्रीमध्ये काय चालू आहे ते आम्हाला माहिती पाहिजे.
याबद्दल
त्यामुळे
एज्युकेशन इन्स्टिट्यूटचे आम्ही आभारी आहोत, छोटे इंडस्ट्रियलिस्ट. ह्या प्रकारे जर एज्युकेशन इन्स्टिट्यूट विचार करायला लागल्या तर इंडस्ट्रीसाठी पण ते चांगलं आहे. इट इज गुड साईन.
हा, पण तुम्ही छोटे इंडस्ट्रियलिस्ट कसं म्हणता येईल आम्हाला? आता तुमची इंडस्ट्री छोटी नाही राहिली.
हो, इंडस्ट्री आहे चालू. आता सध्या तरी स्मॉल स्केलच म्हणतोय आपण.
आमच्या मुली प्रश्न विचारतील, स्मॉल स्केलची डेफिनेशन काय आहे साहेब?
हो, सांगू ना सर, सांगू ना. सगळ्या जेवढ्या डेफिनेशन माहिती आहे किंवा जे थोडंफार नॉलेज आहे तेवढं आपण शेअर करू.
त्यांचा पुढचा प्रश्न असा राहील की विश्वाचं टर्नओव्हर किती आहे, तिला स्मॉल स्केल म्हणायचं का?
हा,
असं विचारतील ना मुली आमच्या.
बर.
करूया सुरू.
हा,
करूया सुरू.
हो चालेल ना सर. इट इज
71 लोक जॉईन झालेले दिसत आहेत.
हो. अजून लोक जॉईन होऊन द्यायचे का चालू करायचं?
चालू करूया मग.
होतील जॉईन सर ते. दरेकर मॅडम.
सर स्क्रीन दिसते का अकोलकर सर?
हो सर दिसतीये. येस सर. दरेकर मॅडम, प्लीज स्टार्ट द फर्स्ट सेशन.
A very good morning to one and all present over here. It gives me immense pleasure to extend to you a very warm welcome on behalf of Department of Chemistry, Radhabai, Kale, Mahila, Mahavidyalaya, Ahmednagar, who has clubbed all of us for the national level webinar on the drug discovery and development 2020. Chemistry Department heartily welcome our esteemed speaker, Dr. Vishnu Halnor sir, Managing Director, VBH Fine Chemicals. Also, Department of Chemistry heartily welcome Dr. Mohammad Sheikh sir, Group Leader, New Drug Discovery Division, Okhad. A warm welcome to our respected Principal, Dr. B.K. Karale sir, for the national webinar. Head of the Department, Dr. Hemant Akolkar sir, Organizing Secretary, Dr. Mubarak Sheikh sir, all my colleagues, participants and technical support team, once again a grand welcome for today's national webinar on drug discovery and development 2020. I would like to mention to all the participants that feedback form link will be shared by the end of the session on the chat section of Zoom, YouTube and on Telegram group. It will remain active till 9 p.m. tonight. Feeling this is a request for obtain a e-certificates, and e-certificates will be issued on your email address within 5 days of the webinar to the registered participants who remain present for the entire webinar sessions and submit it successfully. Without further delay, I wish to request Dr. Hemant Akolkar sir, please introduce our esteemed speaker, Dr. Vishnu Halnor sir, Managing Director, VBH Fine Chemicals, Pune. Please, Akolkar sir.
Thank you, ma'am. Good morning, all. Today, we have Dr. Vishnu Halnor sir as a guest speaker for our first session of the second day. Dr. Vishnu sir is now work as Managing Director of VBH Fine Chemicals, Pune. He has a huge industrial experience of 31 years. He has started his industrial career in 1989 as a senior executive R&D at Lupin Laboratory. And then he joined the SOL Pharmaceutical Limited in 94 as a R&D manager. Then he worked as a plant manager in TASC Pharmaceutical Limited. Also, he worked as a deputy general manager in Glenmark Pharmaceuticals from 2001 to 2004. Then he worked as a vice president in Sai Adventium Pharma Limited. Now he is working as a managing director in VBH Fine Chemicals. Also, he has 2 patents on his credit. So because of his huge industrial experience, we have invited him. It's our privilege to invite him as a guest speaker for this webinar. Now, I request Dr. Halnur sir to please share your views regarding new drug discovery and development in this webinar. Thank you, sir. Please, sir.
Thank you, sir. First of all, this is my a great privilege, and it is a pleasure that to delivering my speech or overviews on the drug discovery development. And I'm thankful to Principal Dr. B.K. Kale sir for inviting me. I'm thankful to Dr. Hemant Akolkar sir for giving this invitation and opportunity as a industry representative. Further, I'm thankful to all members of the Radhabai Kale Mahila Mahavidyalaya and organizing team. So, well, we can start this thing. I will give my overview on the drug discovery and development. Basically, yeah. So this is the introduction, and this is my privilege to give the overview on the drug discovery. So first of all, it is the question to us that what is the drug? Drug is a chemical substance of a known structure that cause a change in organism, physiology or psychological when consumed. So drugs are typically distinguished from the food and substances. Basically, food and substance provides the nutraceuticals to our body, and drugs is a medicine which is the chemical substance which can give the changes in the physiological and psychological changes in the human body. So first of all, we should have the questions to ourselves. What is a discovery? So discovery and what is the drug definition? Drug discovery. Drug is the, as I told you, the general definition, and presently new drug definition of CDSCO. So what is a CDSCO? It is a Central Drug Standard Control Organization of India. So drug as a defined is act includes bulk drug substances which has not been used in the country to any significant extent under the condition prescribed, recommended or suggested in the labeling, and thereof has been organized and effective and safe by the licensing authority mentioned under Rule 21. This I am just giving the information. This is informative for the people who they may not be knowing that, undergraduate or postgraduate people, they may not be knowing this kind of a definition or central drug organization. So this is just for the information I am giving it. So basically, a drug discovery is having a 2 phases. Okay, drug discovery phase. So first discovery phase. What is a discovery phase? It is the synthesis and identification of new chemical entities as a potential therapeutic agent. So mainly synthesis is the most important part, and chemists and scientists is playing the major role for developing the NC molecules. Till that we cannot say that is the drug. So that's what the synthesis of the chemicals. Finally, for the synthesis of any chemical, our chemistry remains same. Either that chemical will be having activity or doesn't have the activity, but it is a new chemical entity. So till this we can say it is a new chemical entities. And then next phase is the development phase. So development phase, compound is tested for the safety and efficacy for one or more clinical indications in the suitable formulation and dosage form. So here also again, it is having the development of the doses and study of the safety as well as the efficacy. And then further, if we go for the introduction, the today's scenario, the term drug discovery has become very conventional. As per my opinion, if we call it a drug invention, that will be more appropriate. That is, I feel. So invention, anyway, we want to do the invention. So drug invention, I feel that that is the more appropriate word for the drug discovery. Until a few decades ago, drugs were discovered as a natural products and used as such for the treating diseases. See, we all of us, we know that our ancestors are using these natural products as a drugs. See, even everyone, we know that even turmeric regularly our people are using. So basically, in the drug development or discoveries or invention, we should have the questions to ourselves. See why it is happening, where it is happening, when it can happen. So these kind of questions are more important in the discoveries. So olden age, our ancestors use all these kind of natural products, but they were not knowing that what is the exactly the ingredient which is curing the disease. But they used to give these natural products to the our community, and they're using it. Our means Ayurveda is giving a lot of Ayurvedic chemical, Ayurvedic medicines, but out of that, some plants we may not know, we don't know today also what are the ingredients that plants are containing, that plants are having. So, say for example, turmeric, our old people are using, but they don't know what is the turmeric is having. See, today we know that because we ask that question that why the turmeric is so effective and giving the impact on our this thing or diseases, it is getting cure the diseases. So after later part, we came to know that it is having the curcumin inside the turmeric. So likewise, there are several natural products are available and several plants existing in India or in the globe which can be used as a natural source of the treatment. So further, today we even our scientists draw the inspiration for the natural. Most of the drug introduced in the market are modified or derivatized to enhance the activity of the drug molecule as compared to their natural counterpart. See, in the natural form, that may not be having that much activity, or it may not give the that much result for the curing the disease or for controlling the deterioration of the human body. But if we do the derivatization of the natural products, so its activity is getting enhanced, and that is having a far better results as compared to the natural products. So it is a combination of natural and allopathical medicines. Okay, due to this terminology, drug invention can be justified as a new molecule are invented by scientists in the laboratory. Okay, so further, just for giving the overview, discovery is having a 5 phases. First is the synthesis, and after the synthesis and screening of the molecule, NC molecule, it is going for the preclinical research. And after preclinical research, it is going for the clinical development. This is the third phase of the study. And after that, it is going for the administration department, that is a Food and Drug Administration, for the review and study of the drug, whatever the release after the clinical development. And after approval of the FDA or authorizing agencies, when they approve the drug, or our CDSCO approve the drug, or WHO approve the drug, it goes to the post-market monitoring. So means it is going for the phase 4, and it is a market. It is distributed in the market for the surveillance. Okay. So drug discovery and development, a long and risky pathway, or it is a long way to go for the discovery of the any drug. So initially, a drug in the drug discovery or invention, we have to screen the molecules, and we can have the NC molecule library. We can produce several compounds. So as per the studies and my knowledge, 3 to 6 years, we can have the 5 to 10,000 compound NC molecules we can synthesize in the laboratories, and we can screen them. And after that screening, we can subject that to the preclinical trials. And it is not necessary that whatever the NC molecules we have synthesized in the laboratories, which all the compounds will have the activities. So out of that, I expect that normally the ratio is that only 200 to 250 compounds will get some preclinical triggers or preclinical activities. And from that after completing that study, so this development and this preclinical studies, normally it takes 3 to 6 years, but sometimes it may take more time. But after 6 years, this can be subjected to the clinical trials. So again, 250 compound will not be having that much activities in the clinical trials. So whatever we subject to the clinical trials, out of this 250, if we get around 5 compounds also, that is a great research work. So these 5 compounds after clinical trials, if we get it, out of that, this is subjected to the approval from the drug authority. So from these 5 also, we cannot expect the all 5 100% will have the activities in the compound. So out of 5, we may get around one compound for the approval. And after FDA review and this will release for the phase 4, and then it will be released for the marketing surveillance. So this is the total phase-wise development of the drug, but this is the long pathway. Okay, so further, this is this drug discovery or invention having the following points covered in that one. First of that one is the target identification and validation. Then second one is the hit discovery. Third one is the assay development and screening. Then high throughput screening, lead optimization, pharmacological profiling. Okay, so in this also we can have target molecules when we are going to identification. So there are the 2 types of this thing: classical pharmacology and this reverse pharmacology. So what is the basic difference between the classical pharmacology and reverse pharmacology? Okay, so relies on the phenotypic screening of chemical libraries of the synthetic small molecule, natural products or extracts to identify substance that have a desirable therapeutic effect. Using the techniques of a medicinal chemistry, the potency, selectivity, and other properties of this screening hits are optimized to produce candidate drug. Only after the compounds have been discovered, an effort is made to determine the biological target of the compounds. So this is the classical pharmacology. And reverse pharmacology, also known as a target-based drug discovery. A hypothesis is first made that modulation of the activity of the specific proteins, specific protein target will have the beneficial therapeutic effect. Screening of a chemical libraries of small molecules is then used to identify compounds that bind with a high affinity to the target. The hit from these screens are then used as a starting point of the drug discovery. So basically, what is the classical approach and what is the reverse pharmacology? So in classical approach, we isolate a molecule or a natural product and then check its biological activities on various diseases and then identify the drug candidate. So here, first we synthesize the NC molecules and then we go for the drug identification process. So this is the pathway for that. Functional activity, first we have to check the functional activity, then ligand or drug receptor isolation, then functional studies, biological role and pathophysiology, and then target disease. So here first we are synthesizing the chemical compounds and then we are just screening it and studying it for the clinical studies, and if it hits then we are using for the diseases. So this is the drug candidate, the classic approach to produce the drug candidate. In reverse pharmacology, what is the difference between these things? So reverse pharmacology is a totally opposite approach. In this case, we know the disease. Bioinformatics and molecular genetics, we will give the receptors information, and we can, from the receptor, we can get the ligand or the drug or NC molecules. So that NC molecule first will go for the first compound screening. So in the first compound screening, again, it is not necessary that 100% trigger out we can get it, or 100% activity we can get of the all compounds. So there some compounds may show the 50% or 60% activities in the first compound screening. Then we are subjecting this first compound screening to the high throughput screening, and to find out the lead compound. In that case, we can get the maximum activity of the compound, maybe 100% activity of the compound, or functional activity and biological role of that compound. So, and then we are going for the target disease. So second compound screening, we can get the drug candidate after second compound screening. So this is the basic difference between the classical and reverse pharmacology. Further, elaborating this classical and reverse pharmacology, you see that this classical pathway is quite expensive, time consuming, and numerous bottlenecks are there. We are facing a lot of problems and in the classical this thing, because first we are targeting our NC molecules, we are synthesizing in the laboratories, and then it is subjecting to the identification, then optimization, then preclinical studies, and then clinical studies. So here, say for example 5,000 to 10,000 compound, we may not get 200 or more than 200 or 250 compounds. After clinical trial, we may get 1 or 2 drug candidates at the end of the studies. But in the reverse pharmacology, here we know the disease, we know the target, and accordingly our research is proceeding, and based on that we are getting large scale trials initially in the initial phase itself, and then it is going for the relevant science, then screening studies, then preclinical studies, then clinical studies. So all these things here, efficacy is getting tested, and here chances are there to get more candidates at the end of the studies. So this is the basic difference between the classical and reverse. So after this thing, all this drug identification, here what is the drug target? The drug is basically targeting in our body G proteins, coupled receptors, enzymes, ion channels, nuclear hormones receptors, structural proteins such as tubulin, and membrane transport proteins. So basically drug is targeting these things in our body. So drug usually act on either cellular or genetic chemical in the body known as the targets, which are believed to be associated with the disease. So basically drug is targeting our genetics, this thing, cellular part or genetic parts. Then target identification finds a gene or protein that plays a significant role in a disease when identified therapeutic characteristics are recorded. Researchers use a variety of techniques to identify and isolate the individual targets, such as a disease association, bioactive molecules, cell-based models, proteins, interactions, signaling pathways, analysis, and functional analysis of the genes. For this kind of these things, there are some libraries. The Sanger whole genome CRISPR library and Duolink PLA are excellent source of the for the drug discovery targets. Okay, so these are the identified these strategies: genomics, proteomics, bioinformatics, phenotype analysis, inverse docking. Then target validation. So target validation is very important part of the process. So target is validation is done to identify and select the most useful target among the various shortlisted targets. So we are selecting several targets, but among that we need to select one few targets or target which is having the most active activity, okay, which will be useful in the development of the new treatment for the disease. Researcher analysis and compare each drug target to other based on their ability to regulate biological and chemical process in the body. So the biological and chemical changes in the body, what is happening, that is more important for the researcher. So that's what this validation is very, very important. Validation also confirms that interaction with the drug target result in a desired change in the behavior of the disease cell. So whatever diseased cell are there, so this behavior pattern will change. Once the target has been confirmed and validated, scientists then move further to identify the compound that have an effect on the selected target. So basically, identification, target identification and validation. So this is the process. Basically, this is the process for the identification and validation. Identification of a biological target for the disease involves identification of genes and proteins involved in the disease. Use human genome, identifies the response using a cell-based or animal model, individual or combination of approach used. So in the validation, ensure that the target is involved in the disease. In vitro tests are performed to confirm that changing the target will result in change in the disease. In vivo test performed with the animal to confirm in the vitro and vitro test. So basically, what is a vitro test and what is a vivo test? Maybe our undergraduate people or maybe graduate people, they may not be having the concept clear. Yeah, here just I want to explain this. What is a vitro and what is a vivo? So vitro is the outside of the body or outside of the organism body. And vivo, that can be done in the petri dish or test tube. We are developing the culture and all these things, we are testing it. And vivo, basically it is in the inside the body of the organism. Okay, the result will predict profile of the new drugs and help determine if effect drug can be made. Okay, so further, lead identification is the very important part of this whole process. Lead identification, compounds or substance is one that is believed to have the potential or treat the target disease, to treat the target disease. So this is what the lead identification is very, very important. And for this purpose, we need to have the all studies in place, and then only we can conclude that this whatever lead we are identifying is having a more effect or more impact, or it is quite effective in that particular disease. Research scientists compare known substance with a new compound to determine their likelihood of the success. Then leads are sometimes developed as a collection or libraries of individual molecules that possesses properties needed in a new drugs. Testing is then done on each of these molecules to confirm its effect on the drug targets. Okay, further moving approach to the new drug molecules. See, earlier old techniques was there, and nowadays lot of new techniques are arrived, or lot of new techniques are invented by our scientists, and which is very quite useful for the further studies or these chemical drug discoveries or chemical scientists. Okay, so in old technique, animal model as a human disease was existing, but now presently molecular modeling is a very good technique which give the fastest results for the identification. Natural product of the plant, animal or microorganism in the old technique. And then now presently combinatorial chemistry is available, traditional medicines. So now biotechnology is existing, modification of a structure of known drugs. Then genetics medicines is available now, random screening approach, immunopharmacological is available now. So these are the techniques available now. So further, just I focus on few more new techniques. I will just elaborate these techniques. Molecular modeling. Okay, aided by three-dimensional computer graphics and allow design of a structure based on the new and known molecule. Highly selective targeted compounds are created by enhancing desired properties of known molecule. Then combinatorial chemistry, what is this? Comprise chemical synthetic method that make it possible to prepare a large number of compounds in a single process. So this is very useful. These compounds, this compound library can be made as a mixture, set of individual compounds or chemical structures generated by the computer software. Then further biotechnology. Biotechnology, what is a biotechnology? Scientists can genetically engineer living system to produce disease fighting biological molecules. So this is what the biotechnology. This they are engineering the living system, and it is very much helpful for the identifying the lead candidate. Then immunopharmacology. It helps in identifying biological immunomodeling agent that cause selective upregulation or downregulation of specific immune response. Okay, so screening further, next type is that phenotypic screening. It is a strategy used in the drug discovery to identify molecule with the ability to alter cell phenotype. Animal model and cell based assay are both strategies and used to identify these molecules. So characteristics, phenotype characteristics are trait of an organism that is more important, and it will help for the lead candidate identification. Increase the understanding of the cell signalizing network involved in a disease process, which can lead to a decrease in amount of clinical failure by identifying factor which contribute significantly to an unexpected activity, toxicities, and lack of efficacy. Okay, so now virtual screening. Virtual screening is also very important part. It is a computational technique used in a drug discovery which stimulate real screening by searching libraries of a small molecule in order to identify those structure which are most likely to bind to the drug targets, typically a protein receptors or an enzyme. Advantages of virtual screening are low cost, investigate compounds that have not yet been synthesized. It can be, means you have not synthesized the compound, but you have investigated that compound, but not yet synthesized. It can be used to reduce the initial number of compounds before using the expensive high throughput screening, that is STS method. So it will be helpful because directly if we subject, the cost impact will be very high, and in that case your discovery will hamper. So virtual screening is quite important and useful. The number of possible virtual molecules available for VS is much higher than those available for the STS. The only disadvantage is that it can substitute the real screening. Okay, the type of virtual screening: target based virtual screening or receptor based virtual screening. It predicts the best interaction between ligand against a molecular target to form a complex. During the execution of this target based virtual screening, the evaluated molecules are sorted according to their affinity to the receptor site. Then second one is the ligand based virtual screening or similar based virtual screening. The method uses the information present in the known activity ligand rather than the structure of the target proteins for the both lead identification and optimization. High throughput screening. What is high throughput screening? It is the process of testing a large number of diverse chemical structures against a disease target to identify hits. Using a robotic data processing, control software, liquid handling devices, and sensitive detectors, high throughput screening allows a researcher to quickly conduct millions of chemical, genetic, or pharmacological tests. So that is quite useful. Through this process, one can rapidly identify activate compounds within a short span of the time. Then advantages of the high throughput screening, high sensitivity and speed of the assay, very simple to handle, high information harvest based on the ligand target interaction principle. The technique used for the STS are fluorescence, nuclear magnetic resonance, affinity chromatography, surface plasmon resonance, and DNA microarray. So lead synthesis optimization. So now in whole process, the synthesis is a very, very important part of either or your classical or reverse pharmacology. Without synthesis, you cannot proceed or your process cannot start. The lead optimization of the lead compound obtained through the synthesis and screening compares their properties and provides information to help pharmacological companies select the compound or compounds with the greatest potential to be developed into a safe and effective medicines. So this is what it is giving the more important. Once the compound are shortlisted, lead prioritization studies are conducted in living organism in vivo, which I have just explained in the previous slide. In vivo, that is inside the organism, and in cell, in the test tube or in the other open part, it can be done. That is in vitro. To compare to the various lead compounds to observe how they are metabolized and affect the body. Analogous of the initial lead are prepared and are then tested by the biologist and biological system. Chemists further process this information and data to make additional alteration, which are then further tested on biology. So chemists, by using this information, they can do the alteration in the NC molecules or targeted molecules or identified lead. Further they can develop or further they can derivatize, and which can be having the further more activity or more effectivity than the previous compound. So here, just for the example purpose, I have just quoted this thing, few molecules. Morphine is a painkiller. What is the target of the morphine? Morphine is a targeting your G proteins coupled receptors. Then clopidogrel is an anticoagulant. So what it does, clopidogrel, what it does? It is the targeting the G proteins coupled receptors. So these two molecules are G protein coupled receptor. Just I am giving an example for that. Then erbuline mesylate is anti-cancer. It is anti-cancer drug. So where it is getting coupled, or what it is doing? It is a coupling with a structural protein, that is tubulin. Then fluconazole, it is antifungal, target enzyme inhibition. So these are the few molecules. Just I have example purpose I have quoted it. So studies conducted during the lead optimization. What is this lead optimization? What it includes? Basically, it is the identification of a pharmacore on the lead compound, which is responsible for the biological activities. So that this identification is more important, then it can be subjected for the further studies. Functional group modification. A lead compound helps us in enabling or disabling certain biological effects. Further, structure, activity, and relationship is the relationship between the chemical structure of a molecule and its biological activities. SAR analysis help us determine which chemical group is responsible for showing the desirable biological activities in the organism. Okay, and further structure modification enhance the therapeutic and potency index the lead molecule. Then quantitative structure activity relationship is a parameter which correlates the biological efficacy of a lead compound to its physiochemical properties, such as the electronic effect, steric effect, and lipophilicity effect. Then further molecular graphic based during design to find a structure match, and docking studies. So docking studies is also more important, are conducted, which deals with the ligand conformation, predictions, and orientation with the target active sites. Its prediction, the various force acting between the target and ligands. Okay, then this is the Chris Lipinski rule of 5. You must be knowing all these things. So molecular weight of the lead identified, it it should be less than 500, or H bond donors, hydrogen bond donors, hydrogen bond acceptors. So this should be having in this numbers, and then participating coefficient. So this is for any drug molecule to be orally active, it has to be follow this thumb rule. So for oral drugs, we need to follow this Chris Lipinski rules of 5. Then successful drug must be absorbed in the bloodstream, distributed. Okay, so a proper site of this body metabolize. So this, before these studies, before proceeding further, just I want to explain here. So lead synthesis, NC molecule synthesis. See, at the end of the day, chemistry will be same for all kind of the molecules. Either it is a chemical, or it is a drug, or it is the intermediates, or whatever it is there. So chemistry will remain same. So what is required for the scientist to think? So here I will be requesting all the educational institute, at least you target to create at least one candidate, or one chemical scientist, or one physical scientist in a year from each institute or each college. That will help to generate scientists, or that will help to develop the scientific minds in the our country, or which will be helping for our research. So this is my request for the all educational institutes. So at least you target department wise. So per year you need to at least develop one scientist from each department. After that, if you develop one scientist is a year, it is a great achievement for the educational institute. So here I will just apart from this subject, I'm just going for the some suggestions for the, or some techniques I want to tell to the people which is undergraduate. So after graduation, or the people are professor or lecturer, or maybe PhD students, or postdocs, they may not be require this thing, but undergraduate people, or BSc candidate, or MSc candidate, this will be helpful for them to develop themselves, or motivate themselves, and to approach towards the scientific mentality. So first of all, as per my opinion, any person who, they have to think that, see, what is the scientist in our field, in a chemical field, it is a chemical scientist or chemist. But all chemists cannot be scientists, but all scientists in the chemistry, they can be all chemists. So here, as per my opinion, first of all chemists, they should ask the questions themselves: why it is happening, when it is happened, and when it is going to happen. So this questioning themselves is a more important. So sometimes in a due course of our lifetime, we can see our life stages. So if you see our childs or children are asking several questions, they don't know the answer. They're trying to find the answer for the question. So first they're asking the question. The questions are coming in mind. It is the first sign of the scientist, as per my opinion. So first, you should have the question why it is happening, and what is going to happen, when it is going to happen. So this is very important for the upcoming scientists or upcoming chemists, and they should think about that. Without that, you cannot go for the research work. So your mind cannot go for the research line. So sometimes if you see that after child, you have to have the childish question yourself. You become a child. Okay, even though you are studying in the MSc, no problem. You become a 5-year boy. You ask a 5-year girl. You can ask your question yourself. Why this is happening? Anything you can, anything in your daily life, when you are going to the college or going to the school, you can ask yourself why this is happening. Okay, sometimes we are criticizing some of our teachers or professors. In that also you can have some research. You can ask yourself why this teacher or why this professor or why this lecturer, they are teaching like this. What is the methodology of this one? So this is research is endless process. So for these undergraduate people, they should have the uh, thinking process and dedication if they want to be a scientist. And even MSc people, they can have this kind of approach. Ask the question yourself and try to find out the answer for your question yourself. So that is the scientific approach, and you will get the answer. If you don't get the answer, but at least you can reach nearby the answer. And so far in my whole life, I realized that when we are asking the question ourselves, we are trying to find out the answer for that. And for that, we are doing lot of work, lot of research, lot of studies, or we are approaching several people. We are trying to get the answer. So this is the scientific approach. And I think the young generation should start questioning themselves and try to resolve the question or try to answer the question, get the solution for the questions. So this is most important for the developing the scientific mind. So even professor or lecturer, they can ask the several questions to the students and try to develop the creativity in the mind of the young students or undergraduate student or postgraduate student. So this is what my opinion, and I think some professor or people will definitely or they will support this my opinion. Okay, so further, successful drug. What is the successful drug? When it absorbs in the bloodstream, that is the successful drug. Distributed to the proper site of the action in the body. It has to go to the proper site of the body. Otherwise, when you are taking the drug and it is not attacking the where it has to be, then it is useless drug. Metabolize efficient and effectively. Okay, it should be metabolized efficiently and effectively. It should not retain in our bodies. Okay, it should be excreted from our body successfully, then demonstrated to a not toxic. So this should be safe drug. So it is basically the conversion of drug candidate to the drug product for the human clinical trials. Lab and animal testing to determine if the drug is safe enough for the human testing. So drug has to be safe. So that is what our clinical study is required. Testing the lead compound extensively to determine if they should move on to testing in human. Scientists carry out in vitro and vivo tests. Scientists try to understand how the drug work and what the safety and profile looks like. Aim of a preclinical testing. What is the aim of that? That is mainly whatever the point I have explained. That is safety of the drug or safety of the pharmacology is very, very important. So pharmacological testing and check that drug does not produce any hazardous side effects. So that is most important. Otherwise, any drug is curing your disease, but it is creating a lot of side effects. So again, it is a very harmful. Then formulation, preliminary toxicological testing or eliminate genotoxicity and to determine the maximum non-toxic dose of the drug. Animal studies, pharmacokinetic studies on absorption, metabolism, distribution, and elimination in a lab animal like mice, chicken, monkeys, and guinea pigs. Okay, components of the animal studies, toxicology studies, mutagenicity studies, carcinogenicity studies, reproductive studies. These are the important factors, important parts. Chemical and pharmaceutical development. What is this? Feasibility of large scale synthesis and purification. We need to check the feasibility. Otherwise, if we go for the large scale and if it is not feasible, then again it is useless. Then stability of the compound under various conditions. That is very, very important part of this one. Otherwise, if the compound is not stable, after taking the drug, it can, if it is not stable, it can create a hazardous side effects, and that will be harmful for the any human bodies or any organism. To develop the suitable formulation. So this is develop the suitable formulation is also one of the dose formation and studies. So limitation, what is the limitation of a preclinical study? Toxicity study is the consuming and expensive. This is the limitation of this preclinical study. It is taking 4 to 6 years, may be required or collect and analyze data on toxicity for testing in humans. So this I'm just quoting the standard time. Sometimes it may take more than 6 years also. Large number of animals may be needed to obtain the valid preclinical data. So see, to get the preclinical data, we may require a large number of animals. And again, it is a big problem or big question mark. It raises the ethical issues. Okay, the human means animal rights or human rights or this personal this thing, because we are killing the animals for the studies. So then extrapolation of the therapeutic index and toxicity data from the human are reasonably predicts for the many but not for all toxic. For the statistical reason, real adverse effects are unlikely to be detected in the preclinical testing. Okay, so now coming to the good laboratory practices, there are 5 fundamentals: resource, characterization, resource. In the resource, what is there? Personnel are there, more important. Personnel is the chemist and scientists, facilities and equipments. These things are required. These resources are required. Characterization. For characterization, for identification and all these things, we need this characterization. Then rules. What is the protocol and procedures? Results. Then quality assurance. So these are the 5 important factors for the laboratories. So coming to the final conclusion. So preclinical studies and clinical studies. So this is the total conclusion of my overview. It is a discovery. Chemistry, pharmacology, search for the active substance, toxicology, efficacy studies on various types of the animals. So first, synthesis, the new chemical entities, then subjecting to the studies, and then efficacy studies. Efficacy study is very important. Then it is subjected to the IND. So investigational new drug application for the permission to administer a new drug to human. So this is the regulatory, this thing. And then it is further, it is going for the early clinical studies. That is the phase 1. So in the phase 1, efficacy studies on healthy volunteers. So maybe number 50 or 100, maybe some people may say that their number may be different. But these are the healthy volunteers for the phase 1. They are not patients. Then phase 2 is a clinical studies on the limited scale, that is 100 to 200 patients. But here the difference is that these are the patients. These are not healthy persons. Then it goes to the phase 3 studies. Here the number of patients will be more. And then after successful clinical studies, here after this phase 3, it is not necessary that you may get your lead candidate successfully go to the NDA, or it may stop at the phase 2 clinical trials also, or phase 3 clinical trials also. So success ratio, it depends on our initial studies and development. Then it goes to the NDA. What is NDA? NDA is a new drug application. Okay, so application for the permission to market a new drug. And then it goes to the phase 4. And what is the phase 4? Continued comparative studies. What is the phase 4? It is going to the market and surveillance. So these are the studies. So this is what my overview. Thank you very much. And here I want to just give some few notes on my this thing. Presently, I am in a generic business, API company. We are having the API company. We are producing generic APIs, and I'm having 2 companies. One is VBH Fine Chemical. That is my proprietary company, and Vishwa Laboratories is a private limited company. So this company I have started in 2014, and commercial manufacturing started from 2017. So here my main point to tell you people that I'm having the target down the line 1 year. I'm going to start the research laboratory which is under construction. So here I'm going for the custom research or new chemical entity research very soon. I'm planning to create a facility that is sufficient to work for the minimum 25 scientists or maximum 50 scientists. So that is what the work is under progress, and maybe before 1 year this activity will be started. So thank you very much. So here, first of all, I'm very much thankful to Dr. B.K. Karale giving me this opportunity to just give my thoughts and overview on the drug discovery, an important topic. And then as an educational institute, they have prepared the industrial resource, or they thought that this industrial resource will be helpful to develop a scientist. So this is my request to Dr. Bhausep Karale, Principal Dr. Bhausep Karale sir, to develop some scientists in a shortest time from your institute. That will be great work. So thank you very much. Thanks to all participants. Thanks to all faculties. Thanks to all organizer committee. And finally, again, thanks to Dr. B.K. Karale sir. Thank you, sir. Over to you.
Thank you, sir. Thank you very much. Dear participants, if you have any queries, doubts, then please ask or text in chat box. Sir, there is one question.
Yeah.
In your Vishwa laboratory, which type of drugs are synthesized? Which method are used in laboratory for the drug production?
Yeah.
Question is asked by Suhas Pathare.
Yeah. Presently, we are manufacturing anti-inflammatory drugs, anti-coagulant drugs, and main classic drug is anti-vitiligo. That is what the white patches is coming on the skin for that. And one is anti-musculant. Then we are having anti-emetic drugs and antiviral drugs. So antiviral in vitro HIV drugs also I'm manufacturing. And here we are having all conventional methods we are following, we are developing, and for the cost effectiveness, we are having the laboratory support, process development lab in place. Yeah. Further, any question?
Sir, there is another one question. What is total cost to develop the drug candidate? Question asked by Dr. Satish Birajdar.
Yeah, but drug development cost, it depends on the length of the drug and the time required for the development of the drug. So it is quite high cost for the development of the drug, which is means quantification is depend on the drug size or length of the drug synthesis. So approximately 1 to 1.5 billion is the approximate costing is required for the one drug development. That is my this analysis. Around 1 to 1.5 billion dollars.
Okay, sir. Thank you, sir. There are many appreciating comments and messages are being seen in the chat box of Zoom and YouTube channel.
Yeah,
thank you, sir.
Thank you very much.
You have very nicely explained overview on the drug discovery and development. You have explained phases of drug discovery and development, basic difference between classical pharmacology and reverse pharmacology. You have explained what is the target identification, target identification strategies, target validation, lead identification, lead synthesis optimization, preclinical development. So thank you, sir, for sharing such valuable information about drug discovery and invention. Thank you very much, sir, for such an insightful session. Thank you, sir.
Thank you, madam. Thank you very much.
Okay, now let's start our second session. Once again, welcome one and all to the second session of webinar. Now I request Dr. Mubarak Sheikh sir to introduce our esteemed speaker of this session, Dr. Mohammad Sheikh sir, Group Leader, New Drug Discovery Division, Okhad, Aurangabad. Please, Dr. Sheikh sir.
Thank you, Director Madam. Good morning to all respected dignitaries and all the delegates. I am Dr. Mubarak Sheikh. First of all, I would like to appreciate your active participation in today's webinar on Drug Discovery and Development 2020. So after excellent presentation by Dr. Hanur sir, now it's my privilege to introduce today's our another speaker, Dr. Mohammad Sheikh. Dr. Mohammad Sheikh currently working as a group leader in New Drug Discovery Division in Okhad Research Center at Aurangabad. So uh, he is he completed his PhD from Dr. Babasaheb Ambedkar University, Aurangabad in 2010. And fortunately, he is our alumni rare student. He completed his BSc and MSc from the RBNB College, Srirampur. He completed his MSc in Organic Chemistry in 2000 from the RBNB College, Srirampur. He also So after completing his MSc and PhD, he joined to the industry. So he started his industrial career from the Harman Phenochem as a chemist in the process development division from 2001. Then he switched to another company, that is Suven Life Sciences, there where he worked as a senior research chemist for the 2 years. And after that, in 2004, he joined to the Okhad Research Center, and now presently he is working as a group leader in the Okhad Research Center in the New Drug Discovery Division. So he is expertise from 18 years. He is expertise in the new drug, designing the new drug discovery, and his expertise in the synthesis of new chemical entities for biological evaluation for target-based generation of new leads for targeted diseases. So while working from the last 18 years, to his credit, 5 years US patent are there, and also along with this 15 research articles to his credit. So from them, European Journal of Medicinal Chemistry, Bioorganic Medicinal Chemistry, Tetrahedral Letter, Chinese Chemical Letters, Journal of Heterocyclic Chemistry. These are the some predominant research articles to his credit. So such an eminent personality with us to share his experience from the industry. So I welcome Dr. Mohammad Sheikh sir. Please share your opinion and guide to us. Thank you. Sheikh sir.
शेअर तुमची केली सर शेअर तुमची केली स्क्रीन
Very good morning to all of you.
Very good morning, sir.
Thank you, Dr. Mubarak Sheikh, for nice introduction. I am very much thankful to Dr. B.K. Karale sir for giving me this opportunity to talk on this webinar. And I am also thankful to organizing committee, Dr. Akolkar sir, Dr. Mubarak, and all the committee members. Today, I am here to present role of Indian pharmaceutical companies in the development of new drug molecule. My presentation contains introduction to the drug discovery, role of Indian companies, pipelines of Indian companies, and conclusion. What is drug? A chemical substance of known structure other than a nutrient or an essential dietary ingredient which, when administered to a living organism, produces a biological effect. Any substance or product that is used or intended to be used to modify or explore the psychological system or pathological state in the benefit of the recipient. Any substance or product that is used in the treatment, cure, prevention, or diagnosis of disease or used to otherwise enhance physical or mental well-being. Simply, we can call that chemical substance used as medicine known as drug. Difference between branded versus generic drug. Branded drugs, we called as innovator drug. Branded drug is the product that is patented by inventor company. Branded drug can be produced and sold by the company holding patent. Branded drugs are costly to cover the R&D expenses. Too much R&D expenses to discover a single molecule. And on the other hand, generic drugs. Generic drugs comparable to branded drug in dosage from strength, route of administration, quality, performance, and intended use can be produced and sold after original patent expire. Generic drugs are cheaper, less developmental cost. What is drug discovery? Drug discovery is the process through which potential new medicines are identified. It involves a wide range of scientific disciplines, including biology, chemistry, pharmacology. Drug discovery is the process by which new candidate medications are discovered. Once a compound that fulfills all of these requirements has been identified, the process of drug development continues. Stages of new drug synthesis. Previous presentation, Harnold sir cover the stages. In the stages of new drug synthesis, total 3 stages are there: drug discovery, preclinical development, clinical development. Drug discovery. Candidate molecules are chosen on the basis of pharmacological property. Preclinical development. Non-human studies, that is toxicity testing, pharmacokinetic analysis, and formulation are performed. Clinical development. The selected compound is tested for efficacy, side effects, and potential dangers in volunteers and patients. Stages of process of drug discovery. There are 5 stages. Stage 1, discovery and development. Stage 2, preclinical research. Stage 3, clinical development. Stage 4, FDA review. And last, 5th stage, marketing. History of drug discovery. In early 90s, 1919, herbal drugs and serendipitous discoveries. In 1920s and 30s, vitamins and vaccines. In 1940s, antibiotic era. R&D boost due to World War II. In 1940s, due to World War II, the R&D development are boost. In 1950s, new technologies and discovery of DNA. In 1960s, breakthrough in etiology. Etiology is to, to break the cause of disease. In 1970s, rise of biotechnology and use of information technology. Now, in 1980s, commercialization of drug discovery and combinatorial chemistry. Then late 90s, 1990s, robotic and automation take place. Why R&D costs are very high? Because challenges of drug discovery, finding one molecule that meets multiple criteria, 10,000 drug candidates to up to the one single drug molecule, the different factors affecting to develop a single drug molecule. That is, the drug candidate should be potent, having potency, having selectivity, selective, reversible, targeted, soluble, long lasting, metabolic, stable, permeable. Up to this stage, the candidates are developed. The major factor was toxicity. The candidate should be non-toxic, non-mutagenic, non-teratogenic. Teratogenic, that means the defect from birth or defect by birth are avoided. That is non-teratogenic. Physically stable, the drug should be patentable and manufacturable. These are the challenges of drug discovery, finding one molecule that meets multiple criteria for develop a single drug molecule. Desirable profile of new chemical entity. Profile of a new chemical entity depends on integrity, API stability, log-P, solubility, pKa, permeability, CYP50 inhibition, metabolic stability, blood-brain barrier. These are the factors of profile of a new chemical entity. Further test for a new chemical entity to become a drug. In vivo efficacy in animal, the major part of drug. Pharmacokinetic and pharmacodynamic studies, safety in animals, formulation studies, levels in healthy subjects, efficacy in patients, ethnic groups. Drug discovery, a multi-step process. New chemical entity synthesis is the primary step, which can be further the synthesized compound or library of compounds screened in vitro and in vitro in vivo analysis for lead optimization, which can be further take for preclinical toxicology. This is multidisciplinary discovery phase. It will take around 3 to 4 years and cost around 20 years, which can be further goes in clinical trials, phase 1, phase 2, phase 3. That is IND, investigation, new drug. But in between preclinical toxicology and clinical trials, applied research is there. That is analytical development of the compound which was analyzed by analytical techniques and their studies. Then scale up of the synthesized compound. Then manufacturing and dose form of the synthesized drug. This will cost 25% from preclinical toxicology to clinical trials. Clinical trial research from phase 1, phase 2, phase 3 takes around 3 to 6 years, it cost around 55%. The clinical, after completion of clinical trial, the compound enters in FDA review of NDA, new drug approval, that is drug regulatory, and it will take around 2 years. And after that, the compound in fourth phase, that is market. The estimated international drug discovery cost around 300 to 500 million dollars. Reason for failure of drug. There are several reasons for failure of drug. Commercial reasons around 5%, animal toxicity 11%, adverse effect in human 10%, lack of efficacy having 30%, and major part 39% is poor pharmaceutical properties. Role of Indian pharmaceutical companies. Drug discovery research setups in India. There are many drug discovery labs in India. The complete drug discovery setups are computational chemistry, medicinal chemistry, analytical chemistry, process research, biological research, pharmacokinetic and pharmacodynamic, toxicology research, clinical research, and regulatory affairs. Ukhad Research Center, Dr. Reddy's Lab, Ranbaxy, Zydus Cadila, Glenmark, and Orchid having complete drug discovery setups. Custom research organizations, CRO, computational, Surujunjar, medicinal chemistry, Sci Adventium, analytical research, Vimta Lab, process research, Almilo, biological and toxicology, Advinus, and clinical Sigma Labs. Business model in India. Develop drug on its own basis. Development of new chemical entity to the market level. Total risk is taken by company Okhad and Zydus Cadila. Collaborative with other multinational companies. Development of NCE to the preclinical or phase 1 and collaborate with multinational company for further development. Risk sharing is with multinational company. That is, the companies are Glenmark, Dr. Reddy's, and Orchid Pharmaceuticals. Part of knowledge based on service providers. Confined to developmental service as required. Sci Adventium, Advinus, GVK Life Sciences, and Vipta Labs, part of knowledge-based service providers. Scenario of drug discovery program. Work in already established therapeutic area as development guidelines and liabilities are known. Mostly work on weaknesses and advantages over competitors' drugs in the market for the product to be a superior somewhere. Spectrum, toxicity, oral, OD. The main part is classical structural activity relationship development, guided often by computational means. Mostly depend on quicker in vitro test to screen compounds. Compromises on fail early, decisions due to pressure to perform, less culture of interdisciplinary research. Zydus Cadila. The molecules target therapeutic area and stage of development. These are the drugs developed by Zydus Cadila, having different therapeutic area launched, having different therapeutic area and having in different phases. Lipaglykine is anti-diabetic drug launched in 2013 in India, whereas ZYH1 in phase 2, ZYD diabetic and obesity drug in phase 1, and remaining drugs, diabetic, obesity drugs are in phase 1. Ranbaxy RBX molecule RBX 11160 having in category malaria in phase 3, whereas RBX 9841 urinary tract infection having completed phase 1 clinical trials. The main part of the molecules which are developed, 5-6 molecules in different therapeutics area are in different stages. RBX 7796 anti-asthmatic compound completed phase 2 suspended. RBX 10558 in IND stage. RBX 2258 in suspended by SWAT Pharma. RBX 7644 antibacterial drug suspended after first phase. RBX 9001 benign prostate hyperplasia suspended during preclinical studies. Dr. Reddy's lab having 6 molecules in the development stage, having in different therapeutic area and stage of developments. Molecules DRL 17822 in phase 1 clinical trial. DRF 2593 diabetic compound, anti-diabetic compound PPAR phase 3 clinical trial. DRF 1042 oncology compound, no funds for phase 2 by Cleantech. DRF 10945 metabolic disorder completed phase 1. DRF 4158 Novartis to decide on clinical trials. DRF 2725 type 2 diabetes insulin sensitizer, no Nordics suspended trial based on right toxicity. Glenmark 7-8 compounds had in different phases like GRC 3886 anti-asthmatic compounds, trials for COPD approved inconclusive. Many of compounds GRC 8200 diabetes type 2 phase 2 completed. GRC 6211 pain killer illegally suspended trial side effects. GRC 4039 anti-arthritis phase 1 completed. GRC 10693 pain phase 1 completed. GRC 15300 pain phase 1 clinical trial. GBR 500 sclerosis in phase 1 clinical trial. Lupin having 6-7 molecules in developing stage. LL4858 anti-TB compound phase 2 clinical trials. LL3348 antipyrosis anti-psoriasis in phase 2 clinical trials. LL4218 anti-psoriasis in phase 2 clinical trials. LLL2011 anti-migraine in phase 2 clinical trials. Unnamed diabetes compound in preclinical stage and unnamed anti-arthritic compound in preclinical development. Piramal Healthcare having 5 to 6 compounds in developing stage. P276 having mental cell lymphoma in phase 2 clinical trials in USA. P1446 oncology in phase 1 in India and Canada. P1736 diabetes type 2 compounds in phase 1 Netherlands, and many more compounds. NPS 30907 dermatology herbal medicine completed phase 2 trials. Okhar from 1997, Okhar started drug discovery, having more than 145 scientists, out of which 43 are PhD, and around 6,500 compounds were screened as a library, and these 4 compounds are in leading. Molecule WCK771, brand name is leononadifloxacin, category antibiotic for the treatment of methicillin resistance Staphylococci aureus MRSA infections, launched in Indian market as MROC and phase 3 clinical trials in USA, stage of development. WCK2349 nadifloxacin. These are fluoroquinolone class compounds, oral antibiotic for the treatment of MRSA infections, launched in Indian market as MROC-O and phase 3 clinical trials in USA. WCK 4873 nafithromycin, ketolite class of compound, category antibiotic, hospital acquired bacterial pneumonia. Stage of development in phase 3 clinical trial in USA, whereas WCK 5222 zidibactam extended spectrum lactamase inhibitor, ESBL inhibitor in phase 3 clinical trials in USA. Conclusion: drug discovery process is tedious and expensive, where the risk of failure is more. Most of the Indian origin drugs are herbal medicine, where research expenses are less compared to the synthetic drugs. Need to train discovery people from beginning if results are required. Need to develop interdisciplinary team, and training also required for an individual to become a team player. Next 10 years are crucial for Indian drug discovery researchers, as many molecules under final stages of development in many Indian companies will decide the path to go forward or backward. I am very much thankful to Okhad Limited, Dr. Mahesh Patel sir, Chief Scientific Officer, New Drug Discovery, Dr. Vijay Patil, Dr. T. Ravi Kumar, Dr. Satish Bhau sir, Dr. Charan Singh Gill, my guide and my mentor, Dr. Prasad Deshpande. Thank you so much. I am very much thankful to Dr. B.K. Karare sir to give me this opportunity to talk on this national webinar on drug discovery and development. Thank you so much.
Thank you, sir. Dear participants, if you have any doubts or queries, then please ask or text it in chat box. Sir, there are many appreciating comments and messages are being seen in the chat box of Zoom and YouTube channel also.
Hello.
Yes, sir, I think there is no question. All are saying it is a nice presentation, excellent presentation. So, thank you, sir. You have gives us detailed information about introduction of drug discovery, difference between branded drugs and generic drugs, what is drug discovery, stages of new drug synthesis, okay, then history of drug discovery, why R&D costs are high. You have given us information about drug discover research setups in India, business modes in India. So thank you very much, sir, for your guidance. Sure, it will help us in future. Once again, thank you, sir. Thank you very much.
Thank you so much to all participant seniors, all the college professors, and all research students and industrial colleagues. Thank you so much.
Thank you, sir. Now let's start our third session of the day. Once again, welcome one and all present over here. May I request Dr. Rupali Endait Madam, please introduce our esteemed speaker, Dr. Narendra Joshi sir, Chief Scientific Officer at Supriya Life Sciences Limited, Mumbai. Please, madam.
Thank you, madam. Good morning to all of you. I take this privilege to introduce you all to the highly experienced and award-winning R&D head with an experience of nearly 29 years in the field of pharmaceuticals and chemical industry, Dr. Narendra Joshi. He has pursued his PhD under the guidance of Professor Dr. C.H. Gill. His expertise in business development strategy, product portfolio selection development pipeline, business planning, business development, as well as organic synthetic chemistry, green chemistry, enzymatic conversion, halogens and fluorochemistry, polymorphs development, steroids, cephalosporins, agrochemicals, etc. He has worked as manager and senior officer in R&D department of reputed companies from 1989 to 2001. He has worked as general manager in process research department in Glenmark from 2001 to 2007. He has worked as director R&D department in Innogent Laboratories Private Limited, Hyderabad from 2010 to 2011. He has also served as vice president R&D department in Amoli Organics Private Limited and Gujarat Fluorochemicals Limited, Baroda from 2011 to 2016. Then from 2017 to 2020, he worked as the head at Aquafarm Chemicals Private Limited, Pune, and Luxai Life Sciences Private Limited, Hyderabad, at the Synthesis and R&D department. Presently, he is Chief Scientific Officer at Supriya Life Sciences, Maharashtra. He has received Best Scientific Contribution to Pharmaceutical Industry Award at Golden Global Health and Education International Conference, Dubai. Excellent contribution to API process development and manufacturing by LOQMAN Awards, supported by Institute of Entrepreneurship Development, Government of UP. Outstanding contribution to API process research award by Pharmaceutical Royal International Society in Bhopal. Received 100 Most Impactful Healthcare Leaders Award by World Health and Wellness Congress. With his brief introduction, I once again welcome Dr. Narendra Joshi sir and request him to deliver his talk. Please welcome sir, Dr. Narendra Joshi.
Yeah, thank you very much, Dr. Rupali, for my introduction.
Thank you, sir.
I will start
Welcome,
now. Slides you will attach there, or I have to upload?
Sir, you can share
please.
screen.
Yeah, just a minute, then I will. Can you attach from your side? Because I forward it to you, or?
Yes, sir. You can forward it to me at my mail. Okay.
Because it is not in my desktop right now, but I will just try. Just a minute. Just wait for 1 minute. Huh? Yeah, good morning, all of you. So thanks for giving me this opportunity to explain my knowledge in field of R&D and drug discovery. Thank you, Karale sir and his team. Now, all Dr. Vishnu and Dr. Sheikh has explained about all drug discovery. So I will go run through all basic definitions, and I will just explain you how organic chemistry and medicinal chemistry is useful for the drug discovery. And I will also let you know what is the market like potential for drug discovery currently in India, as well as process chemistry means generic drugs, and what are the cycle, because Dr. Sheikh has already explained all this, but again I will explain that. So now drugs, as you understood, that it is basically a chemical substances that can change your body and your mind work. They include prescription medicines, over-the-counter medicines, alcohol, tobacco, illegal drugs. So this all comes under drugs. And if you see medicines are a drug which used to diagnose, cure, treat, and prevent diseases. So I explained on earlier slide, there are classification of drugs. It's a prescription drug and over-the-counter drug. So all you must be knowing prescription drugs which are controlled by our FDA and CDSCO, because without prescription, several medicines are not supposed to issue to everyone. It is on under doctor prescription only it should be given. Over-the-counter drugs are easy to get. We can go to a medicine shop and we can get that. And in the classification, like I want to explain more on this, on the small molecule drugs means all chemicals. This is usually derived by chemical synthesis, or it is designed by like matching with natural products. And other field which is biopharmaceutical, which is growing a lot in terms of human treatments, which is recombinant proteins, vaccines, gene therapy, monoclonal antibodies, and cell therapy. This is growing a lot now in over world. So I'll explain what is organic chemistry, because everyone must be studying in organic chemistry with our college.
Sir. Hello, sir.
Yeah.
Sorry to disturb you. Sir, slides are not visible, sir.
Slides are not visible?
Yes, sir. Yes, sir.
But I loaded that. Just a minute.
Yes.
Screen sharing. Slide share.
Sir, please share a slide screen. Share your screen, sir. PPT screen.
It is now.
Yes, sir. Yes, sir. It is now visible, sir. Yes, sir.
Now visible? Okay.
Yes, sir. Yes.
Sorry for inconvenience. Yeah. Now on organic chemistry, organic chemistry, which I also studied in organic chemistry from Raj Shikshan Sanstha up to BSc and MSc from Ahmednagar College, and PhD also from Pune University under guidance of Dr. C.H. Gill. Dr. Rupali also explained. Now, organic chemistry is a branch of chemistry that studies the structure, properties, reactions of organic compounds which contain carbon in covalent bonding. Study of structure determines their chemical composition and formula, and study of properties includes physical, chemical properties, and evaluation of chemical reactivity to understand their behavior. So basically, I want to explain organic chemistry because in India, majority all chemical companies and pharma companies, API companies are basically producing generic drugs. So organic chemistry is very useful in that field in terms of process chemistry, development chemistry, upgradation in terms of technologies available for old chemicals. And in organic chemistry, the study of organic reactions includes the chemical synthesis of natural products and drugs and polymers. So polymers also requires very good knowledge of organic chemistry. And other part of chemistry which majorly required for drug discovery, where knowledge of medicinal chemistry is required. So medicinal chemistry and pharmaceutical chemistry are disciplines at the intersection of chemistry, especially synthetic organic chemistry and pharmacology are the combination in that. So pharmacology, in the next slides, I will explain what are the pharmacology knowledge is required for drug discovery. So pharmacology and various other biological specialties are required in studying medicinal chemistry. Majorly chemical synthesis is the artificial execution of useful chemical reactions to obtain one or several products. This is a chemical synthesis is very much required in designing and synthesizing any chemical substance or NCE. So NCE in all this you understood in the previous presentations. And a chemical synthesis involves the reagents or reactants that will undergo a transformation when subjected to certain conditions. Various reactions types can be applied to formulate a desired product. This requires mixing the compounds in a simple round bottom flask. Many reactions require some form of workup or purification procedures to isolate the final product. So this all you must have studied in your post-graduation syllabus. And this part in chemical synthesis is very important. So I was explaining NCE. NCE is a new chemical entity which gets as a preclinical candidate for drug discovery studies. So NCE is what? NCE is the active moiety is a molecule or ion, excluding those appended portions of the molecule that cause the drug to be an ester, salt, or other derivatives of that molecule, which is responsible for the physiological or pharmacological actions of the drug substance. And an NCE is a molecule developed by the innovator company in the early drug discovery stage. So NCEs are early drug discovery stage chemicals, and here medicinal chemists and organic chemists are required to develop the NCEs. So for designing NCEs, structure activity relationship is very important. That is getting taught in medicinal chemistry subjects. This is the structure activity relationship is the relationship between the chemical or 3D structure of a molecule and its biological activity. So biological activity analysis, there are various databases, softwares are available to see the compatibility of our organic structure with a particular enzyme or particular active cell. So the analysis of SAR enables the determination of the chemical groups responsible for evoking a target biological effect in the organism. This allows modification of the effect of the potency of that bioactive compound by changing the chemical structure. This method was refined to build mathematical relationship between the chemical structure and the biological activity, known as quantitative structure activity relationship. So quantitative QSR studies are very much required while designing the NCE in discoveries. So in India now, if we'll see drug discovery and generic drugs or off-patented drugs. So majorly India is very strong in producing generic drugs in terms of API, in terms of formulation, and we are in the world we are on the second position. But unfortunately, on drug discovery, we have to go far. And in drug discovery, there are very few companies. As Dr. Sheikh has shown, there are only 7-8 companies has reached very few molecules in the phase 1, phase 3 stage. But now India is more focusing or more advancing in biopharmaceuticals and vaccines. So in drug discovery, I would like to take you to understand I explained you what is the structure activity relationship, but now what are the targets? What is target? What is target? So target is the naturally existing cellular or molecular structure involved in the pathology of interest where the drug in development is meant to act. So in SAR studies, this target is getting used to study whether our chemical is active for particular that cell or particular that biological part. So screening and design. So after that targets, we have to screen our synthesized molecule, and based on that, you can modify the functional groups which can activate that molecule. The process of finding a new drug against a chosen target for a particular disease usually involves high throughput screening that STS also explained to you by Dr. Vishnu. So a major part in drug discovery is lead optimization. So lead identification, you understood from last two Dr. Vishnu's slides. Lead identification and optimization is one of the most important steps in drug development. The chemical structure, the lead compound is used as a starting point for a chemical modification in order to improve potency, selectivity, or pharmacokinetic parameters. Once a molecule is identified, the next step is to check its ADMET, means absorption, distribution, metabolism, excretion, and toxicity. So these properties need to be studied. So earlier, Dr. Sheikh also explained out of 10,000 molecules, only one becomes a drug. So 10,000 also it's a preclinical. But before that, like from 1 lakh, you get a 10,000, and from 10,000, only one becomes a successful drug. If the molecule has no toxicity and no mutagenicity either, it has potential for as a lead molecule. So you have to check whether what is the toxicity and mutagenicity. Mutagenicity means it's all cancer generating properties. So after determining these activities on the molecule, then you have to go, after finalizing the molecule, then you have to go for in vitro preclinical drug testing. So in vitro means it is in the glass testing. It is not on the cell or on animals. So in vitro testing to be done of any chosen molecule. So this definition is in Latin, it is vitro means it is in the glass. And when live cells are removed from the organism and tested in an artificial control environment, it is called as in vitro testing. And then in vivo preclinical drug testing is it is on the whole body, means on the animals, or performed on the live environment of a whole body, usually on a live animals such as murine models. So after doing that, after finding the molecule, after in vivo and in vitro, you have to develop a pre-formulation. So why I'm explaining here up to all the activities, because from like organic chemist role, medicinal chemistry person's role, then like biological testing means like our B farms, MSc biotechs, they have to work on this in vivo, in vitro testing. So which are the educational streams are getting useful for drug discovery activity, that you will explain, you will understand from my explanation. So after doing in vivo, in vitro, that company need to do the pre-formulation of that chemical or that active moiety, because without formulation, drug cannot be given to the even animals or to the humans for further clinical studies. So pre-formulation is a group of studies that focus on the physicochemical properties of a new drug candidate that affect the drug performance and the development of dosage form. Every drug has an intrinsic chemical and physical properties which has been considered before development of a pharmaceutical formulation. So these physical properties to understand, and that organic chemist is very expert in understanding these properties which are very important for pharmaceutical formulation. Objective of pre-formulation study is to develop the elegant, stable, effective, and safe dosage form, because without dosage form, any chemical cannot be injected or taken by mouth to humans or to the animals. So now Dr. Vishnu also explained what is IND. IND is investigational new drug. So I explain you what is the definition of investigational new drug. So the term investigational new drug refers to a drug developed by a pharmaceutical or biotech company or other organization that is ready for clinical trials on humans. So after filing to the FDA that IND, then they give the permission to go for clinical trials on humans. When a drug reaches this point, the entity submits an application to get the consent of the FDA, means Food and Drug Administration, to begin these trials. This step is important as an approval for the application allows a sponsor to ship the drug across the country to begin testing. So after IND application, one can go for clinical trials. So after IND, there is a new drug application, new NDA. NDA is a comprehensive document that must be submitted to the US FDA or Indian FDA in order to request approval for marketing a new drug in the United States. Drugs for which NDA are submitted will have already passed through several clinical trials. So I will explain in the next slides all the clinical trials. So this is the drug discovery cycle. What like I was explaining, like any compound collections you have made. So you have to do primary assays, in vivo assays, then secondary assays in vitro, in vivo, and then that molecule goes to the SAR studies, and from SAR then again it comes for the design, then again chemical synthesis. So it is a cycle because not a single molecule immediately becomes a preclinical model or preclinical compound. So this need to go through this cycle of understanding its activity, how it is activity is increasing in the in vitro, in vivo studies based on SAR. The SAR gives us the idea of modifying that molecule wherever is required. That way, in this cycle, molecules are or drug discovery chemists are working in the laboratories. So I explained, and earlier Dr. Vishnu also explained, this is the drug discovery to drug development and to file the NDA. This is the activity I will show. Like first, target discovery is a target identification, assay development, bioinformatics, in vivo validation. So before this target discovery, organic chemist has to play a role to find out the molecular libraries. So that molecular libraries are getting screened through in vivo studies, and that after synthesizing the molecule after earlier studies, then it takes at least 2 to 3 years to complete this study. Then it goes to lead identification and lead optimization. So lead identification is the assay development, high throughput screening for hit molecule, then medicinal chemistry, combinatorial and structure based. So this also around lead identification takes 1 year. Then lead optimization, it takes around 1 to 3 years. In that lead optimization, medicinal chemistry, SAR means structure activity relationship development, improve potency, in vivo testing, and all toxicologies need to be studied in the lead optimization. After that, then that becomes a clinical candidate. What we discussed, after this clinical 10,000 clinical candidates, then one becomes a final drug. So preclinical is a then preclinical is toxicity on humans, no, sorry, on animals. And after toxicity and its effectivity understood from animals, then it gets filed as an IND. IND I explained to you earlier. So after IND, then clinical trial starts. So that phase 1, phase 2, phase 3 goes on humans, and and before that phase 1 starting, we need to develop its proper formulation, pre-formulation we can say. After going through phase 1, phase 2, then that formulation modification also wherever needed modifications, it is get done. So phase 1, phase 2, phase 3, all like if you see, it takes several years. So after phase 3, majorly gives the large safety studies, large efficacy studies, large scale measuring then starts at the phase 3, for that API as well as the formulation. And then NDA gets filed, and after NDA get filed in the FDA, then FDA review happens, then registration happens. That also takes 1 to 2 years, and after that, then marketing starts on that. So after that, it becomes a branded molecule, so branded compound or branded formulation. So earlier, Dr. Vishnu explained what is branded and what is generic. So this after this, it becomes a branded formulation. And once we file the clinical candidate, we understand the clinical candidate, pharmaceutical companies files the patent. So once they file the patent, that patent as a new NCE, they file as a NCE. Then after preclinical studies, after phase 1 or phase 2, they file for the indication also. So meanwhile, in the interim, they file the formulation patent also. So companies has to protect their innovation by doing these protections at the different stages, and that way this molecule will get protected for as a NCE. Patent also has a 20 years expiry. So from NCE to market, and then afterwards formulation patents, indication patents, then companies get at least 20 years protection. So after that, after completion of 20 years at that stages, then other companies can be able to produce that drugs and market it. So once that patent expiry happens, then it becomes a generic. So branded and generic is the difference of only a patent expiry. There is no reduction of any activity. So generic drug, I will explain again. This is a medication created to be the same as an existing approved brand name drug in dosage form, safety, strength, route of administration, quality and performance characteristics will be very similar. So generic drug, there are myths like generic drugs are less effective. It is not the truth. Generic drugs are very similar. Only it is off patent drugs. Only the difference is that. So this I was explaining earlier, formulation. So after formulation development, then you have to file the NDA for making that molecule generic. So for generic market, you have to file the ANDA, not NDA. So abbreviated new drug application, it's a application for selling the generic market by your formulation method. Only efficacy in all this, what I explained earlier, should be the same. So I will explain this. Like there are top multinational companies, their expenses on the R&D are very much more than $1 billion. So if you'll see, there are top 15 companies, those are very strong in drug discovery with their expenditures, with their success. But as Dr. Sheikh explained, very few companies, 4 or 5 companies in India is investing on drug discovery, and that investment is also very less if we'll compare with this multinational company. But now on biopharmaceutical, India is going very aggressively and succeeding. So I mean to say, like drug discovery, how it is important for India to grow our pharma market, need to be evaluated by all our scientific bodies and the government, and we should focus more on drug discovery in chemistry as well as in biopharmacy. Thank you very much.
Thank you, sir. Dear participants, if you have any queries or doubts, please ask or chat it, text it in chat box. There is one question. In some cases of drug overdose, which causes chemical toxicology in our body, My question is, after overdose, any other drug is available to inhibit or decrease this toxicity?
Yes, there are several drugs are available like that, because several cases poison also we take. Poison is also a chemical substance, and for that the antidotes are available. Even if you see the example of sodium cyanide, for sodium cyanide there is antidote. So various products are having antidotes, and that are known in the literature and known in the medicine experts. It is definitely available.
Sir, there is one another question. Why we are lacking in drug discovery research in India?
Yeah, because we are lacking because of our financial muscle only. Because as we explained, for drug discovery to making it as a uh, generic or branded drug, it requires a lot of research activity, lot of expenses, and Indian pharma companies are not that strong financially. Now, if you see the example, Lupin, Glenmark, they also could not withstand that financial pressure. And but now, good thing is that, like, we are uh, very good service provider, and several multinational companies are coming in India for drug discovery work. So there are various contract research companies like GVK, Sai Adventium. There are plenty of companies are available. Those are working on giving services. That Dr. Sheikh also mentioned in one slide, like services providers in India are getting increased, and we are getting a lot of business in that segment. But the discovery, there is, like, companies are minimizing the expenses and reducing the drug discovery work in India right now.
Sir, there are some more questions. Whether herbal medicines needs to go through drug discovery and development process, as herbs are used from 1000 years without side effect.
No. Ayurvedic drugs doesn't require this much of a big cycle of testing. They require only some toxicity studies, and if suppose literature is available, then with that literature also any Ayurvedic doctor can prescribe that medicine. So Ayurvedic medicines are easy to develop, and very less costly in terms of development cost.
Sir, one another. Sir, please explain patent policy. Question is asked by Suhas Pathare.
What is the question?
Please explain patent policy.
Patent policy, like, because every country is having their own patent policy, but there is a patent treaty, what like worldwide it is got accepted, and India is also signing of that. So we obey worldwide all regulations about these patents, not only for pharma but engineering, every part. There is a pharma like patent policy. So in India, like patent policy, because
Please.
there are some differences worldwide, US, Europe. We for the salts, India doesn't grant the patents. That is only the difference in Indian patent policy and worldwide patent policies. There are few differences are there in the patent policies country to country, but majorly all patent once we file, expiry period is 20 years. So that is common for every country, almost every country. But few countries are having little different rules.
Thank you, sir. There are many appreciating comments and messages are being seen in the text box of Zoom YouTube channel. Thank you very much, sir, for such informative session. You have given us a detailed information about drug discovery and development. Thank you, sir, for your guidance. Sure, it will help us in future. Thank you once again, sir. Thank you.
Thank you for giving me opportunity. Thanks to Karale sir and all Karale sir's team. Thank you.
Now it's time for valedictory function. We extend a warm welcome to Principal Dr. S.R. Thopte Sir, SSGM College, Kopargaon, for gracing this event with his presence. I heartily welcome Dr. B.K. Karale Sir. I welcome all the eminent speakers and guests from all over the country from different walks of life have come here to share their knowledge and vast experience with us. Also, I welcome all the participants to this valedictory function. May I now request Dr. Hemant Akolkar sir to introduce our today's chief guest, Honorable Principal Dr. S.R. Thopte sir, SSGM College, Kopargaon. Please, Dr. Sir.
Thank you, ma'am. Today, for this valedictory function, it's our pleasure to have Professor. He is working as a principal in SSGM College, Kopargaon. He did his PhD from University of Pune under the guidance of M.G. Kulkarni sir. Then he, after PhD, he did his postdoctoral research in National Chiao University, Taiwan. And after that, second postdoctoral research in Institute of Chemistry, Taipei, Taiwan. And third postdoctoral research in Forest University, USA, under the guidance of Professor P.K. Garg. He is a visiting faculty in Department of Chemistry, University of Turku, Finland. Then his teaching experience is more than 15 years, and under his guidance, 5 research students have completed their PhD and 1 student is pursuing his PhD. He has completed 4 minor research projects and 3 major research projects which are funded by Department of Science and Technology, New Delhi, CSIR, University Grants Commission, UGC, New Delhi, and Savitribai University, Pune. So he has published more than 40 research papers in reputed international journals. So I heartily welcome Dr. S.R. Thopte sir for this valedictory function. Now I request Dr. Thopte sir to please share his views related to discovery and development. Thank you, sir. Please, sir.
Good morning to everybody. Can you hear me?
Yes, sir.
So good morning to everybody. And let me first of all thanks to the organizer of this national webinar organized by Department of Chemistry, Radhabai, Mahila, Mahavidyalaya, Ahmednagar, to Principal Dr. Karale sir and all other organizers for giving me chance to comment on this seminar in a valedictory function. Before that, just I will go through the Shikshan Sanstha, which is established in 1990 by Karmveer Bhaurav Patel. And last year, we have celebrated 100 years of this Sanstha. In this 100 years, what Sanstha has done, what is called as we planted, hello? Excuse me. So planted a small tree and it has grown into a big tree, like a banana tree, which we have a symbol of Shikshan Sanstha. And in these 100 years, how it is grown, now we have 42 senior colleges. It is also expanded in 16 districts in Maharashtra and 1 district in Karnataka. We have 42 colleges, 17 postgraduate colleges, 438 secondary schools, 8 training colleges, 28 primary schools, 17 pre-primary schools, 68 hostels, 7 administrative offices, and 8 ashram shalas. Total we have 737 branches, and we have total number of employees around 14,000, and the student strength is around 5 lakhs. So this is about the Shikshan Sanstha. And as I said that it was a small tree planted and we have many branches. one of the branches is the Radhabai Mahila Mahavidyalaya, which is organizing this national webinar. It is established in 1989. It is permanently affiliated to the University of Pune, and this college is only established for girls. College is located in a spacious area of 6.8 acres. At present, college is offering undergraduate degree programs in art, commerce, and science. And in art, 6 subjects, which is English, Marathi, Hindi, history, economics, and geography. And it's also offered at special levels, 4 subjects. In science, special levels, chemistry, botany, and general level physics, mathematics, zoology. College is also offering postgraduate degree programs in arts and science, organic chemistry, also running one professional course like a BBA, is a CA. Total strength of this college, girl student, 12,000, and college has done a very good progress in last few years, especially under the able guidance of Principal Dr. Kale sir. And as a effect of this, in last year, college has got A gradation by NAC Bangalore with 3.0 score is CGPA score. So about the seminar, Drug Discovery and Development, DDD 2020. I think I must thank organizers and team leaders for choosing this subject, because we are suffering from last 6 or 7 months with the COVID-19 pandemic. And if we understood what is a drug discovery, what is the different stages, I think all the eminent speakers have shed a light on this, the different aspects. What is the drug? What is the pharmacology? And then what is the formulation in the pharmaceuticals, organic part, synthesis? What are the different aspects of that? And I think this information will be very good for our participants, especially students and new researchers, our colleagues, to contribute something into the COVID-19 development as an antiviral drug, or even as a what is called as a treatment for COVID-19. So I thank all the participants for their lectures, who has delivered nice knowledge to our student participants. And I hope this will be useful to all of us to combat with the COVID-19 in this present situation. I also thank, so overall this national webinar was well organized, and the speakers were chosen nicely. They also did their job nicely. So I thank all the speakers, and also I thank organizers for organizing such a type of a national webinar in this COVID-19 situation. And I should thank again for the organizers to give me a chance at the 11th minute, I can say, to participate in this and have some comments on this national webinar. So once again, thanks to all. Have a good day. Thanks.
Thank you, sir. Thank you very much, sir, for sharing your views with us. Thank you for your constant guidance and support. Thank you very much, sir. Now, I request Dr. Mubarak Sheikh, sir, propose vote of thanks.
Thank you, madam. Good morning, one and Honorable Principal Dr. S.R. Thopte, sir, SSGM Kopargaon, our chief guest of this program. Respected Dr. B.K. Karale, sir, Dr. Narendra Joshi, sir, Dr. Mohammed, sir, ladies and gentlemen, it's my privilege to have been asked to propose a vote of thanks on this occasion. I hope you have enjoyed and benefited academically during the 2 days of this webinar. So before beginning the vote of thanks, I, Dr. Mubarak Sheikh, on behalf of the organizing committee of Drug Discovery and Development 2020, would like to apologize for any shortcomings that you may have encountered. Let me call it fraternity of drug discovery and development here together. And on my own behalf, extend a very hearty vote of thanks to all speakers for gracing your important work and sharing with us your findings and opinion in these 2 days. So I must mention our deep sense of appreciation to Principal Dr. V.S. Shivankar, Sir, Secretary of Raya Shikshan Samstha Satara, for the keynote address and his guidance on the opening day of this seminar. I must mention our deep sense of appreciation to our speaker, Dr. Ron Erande from IIT Jodhpur for his explanation of the anti-malarial drug and anti-cancer drug by the natural synthesis. Further, we are grateful to Dr. Bapu Shingte from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, for demonstrating triazole scaffold and its anti-tubercular and antimicrobial activity with us. I may like to express our sincere thanks to Dr. Sangeeta Jagtap Madam for giving an excellent coverage for discovery and synthesis of anti-cancer drugs. I also wish to express my gratitude to Dr. Vishnu Harnor sir for providing a basic information regarding the development in drug discovery. I am also very much grateful to Dr. Mohammad Sheikh sir for analyzing the drug discovery and development. I am also very grateful to Dr. Narendra Joshi sir for his analysis of drug discovery and development. I would like to take this opportunity to place on record our hearty thanks to convener and principal Dr. B.K. Karale sir for the perfect logistic support and guidance he has extended to all of us at this national level webinar, Drug Discovery and Development 2020. I am also very much thankful to Mr. Vilas Elke sir for all the technical assistance during this 2 days national level webinar, Drug Discovery and Development 2020. Well, an event like this cannot happen in overnight. The wheels start rolling weeks ago. It requires planning and bird's eye for details. We have been fortunate enough to be backed by a team of very motivated and dedicated, our principal, Dr. B.K. Karale sir, our head, Dr. Hemant Akolkar sir, head Department of Chemistry, and colleagues, Ms. N.R. Darekar madam and Dr. R.S. Endait madam, who know their job and are result oriented. I cannot thank everyone enough for their involvement and the willingness to take on the completion of tasks beyond their comfort zones. Respected dignitaries and all the delegates, once again, I want to state that we are all most grateful to all speakers on this stage. Last but not least, I am also very much thankful to all the participants. Without their support, this webinar will not be successful. So we thank you for being with us for this national level webinar, Drug Discovery and Development 2020. It's been a great pleasure. Thank you very much on the behalf of Organizing Committee of Drug Discovery and Development 2020 and the Department of Chemistry of Radhabai, Kale, Mahila, Mahavidyalaya, Ahmednagar. Thank you very much for with us. And so link is provided in the chat box. So there is a note for all the participants, we are sharing the feedback link on the chat box on Telegram group, YouTube chat section. So everyone can fill up that form. The feedback link will be open till 9 p.m. tonight. Thank you so much. And after filling the feedback form, you will get the participant certificate. Thank you. Thank you very much.
Thank you.
Thank you.